Hello everyone, my name is Anson, I'm from the OFK lab, and today I'll be telling you about my project on developing a gene editing nanoparticles as an antiviral to effectively clear cytomegalovirus in donor lungs before transplantation. So cytomegalovirus, or CMV, is one of the most common causes of mortality and morbidity in lung transplant recipients. This is because transplant recipients are likely to be immunocompromised to prevent transplant rejection, and this gives latent CMV inside the donor lungs a chance to reactivate. CMV reactivation can cause a multitude of complications like chronic allograft rejections and injury, bronchiolitis obliterans syndrome, and pneumonitis, which significantly reduces the life expectancy of lung transplant recipients. It is estimated that up to 50% to 90% of the adult population is infected by CMV. So it is very likely that the donor lungs are infected by the incurable virus. And currently, CMV related complications can be prevented by either limiting the use of CMV infected lungs, but this will greatly reduce the availability of donor organs. Another approach is to administer antiviral drugs prophylactically to recipients, but the efficacy of these treatments are decreasing because of a drug resistant CMV becoming more prevalent. And with the development of gene editing technology like CRISPR Cas9, we can now directly target the latent viral genomes to remove the virus from the donor organs. Here's a figure of how CRISPR Cas9 works. Cas9, shown in green here, forms a functional complex with the single guided RNA or sgRNAs, the blue nucleic acid shown here. And once the complex recognizes a PAM motif shown in yellow here, Cas9 will unwind the DNA and allow sgRNAs to screen for complementary pairing. If the pairing between the RNA and the target DNA matches, the complex will initiate its nuclease activity and cleave both strands of DNA, thereby disrupting the genome. So currently, gene therapy payloads are delivered using viral vectors like adeno-associated virus or lentivirus. These are amazing tools, but they pose a few limitations. So first, upon delivery, the virus could trigger immune responses, which can destroy the therapeutic payload. They're also limited in their um, genetic payload capacity, which means if we were to deliver multiple Cas9 mRNA and sgRNAs, we'll have to deliver it with separately with multiple viruses. And because of this separate delivery, it's very difficult to achieve cell specificity and also control the proportion of the therapeutic payload delivered to each target cell. So we believe our nanoparticle technology can not only allow us to mitigate the shortcomings of traditional viral vectors, but also enhance the therapeutic potential of CRISPR-Cas9. Our goal is to engineer nanoparticles that could, one, minimize the immune response during therapeutic delivery, two, co-deliver Cas9 mRNA and multiple sgRNA within a single nanoparticle to target multiple CMV genome sites. Three, to preferentially deliver the payload to lung endothelial cells and monocytes. And four, lastly, house the best proportion of Cas9 mRNA and sgRNA to maximize the therapeutic outcome. So our plan is to first design sgRNAs that effectively targets the early immediate genes of CMV because it has been shown to disrupt CMV reactivation and replication. And these immediate early genes are R36, R37, R38, R122 exon 5, and R123 exon 4. Then the nanoparticles are synthesized by Michael addition and ring opening epoxide reaction, and then mixed with Cas9 mRNA and sgRNAs. The nanoparticles were then self-assembled with different types of RNA, or what we call the multiplex nanoparticles. So here is an image of multiplex nanoparticle taken with the cryo-ETM. And using modular chemistry, we can fine tune the molecular structure of the nanoparticle to achieve high cell specificity and payload capacity. After that, using the ex vivo lung perfusion platform, or the EVLP developed by the Cypel lab, we can perfuse our multiplex nanoparticle in a CMV infected rodent lung to see if our nanoparticle can eliminate lytic and latent CMV. And the EVLP is a platform that allows lungs to exist outside of the host for a period of time by perfusing the important factors for organ survival. This technology opens up a time window for us to deliver our therapy in an organ specific manner before transplantation, which eliminates the possibility of off target effects to other parts of the body. And after the perfusion, the treated lungs will be screened for editing events, the level of viral load, and DNA, and transplant into a CMV negative rodent to determine the efficacy of our therapy. And by utilizing gene editing nanoparticles to target latent viruses in donor lungs, the pool of viable organs can dramatically be increased, while CMV related transplant complications can be simultaneously decreased. So I would like to thank Professor Omar Khan for his guidance and support. I would like to thank Beyond Science Initiative for hosting this amazing conference. Thank you so much for your time, and feel free to reach out to me if you have any questions. Thank you.